So, good afternoon. Thank you, Stephen, for the introduction. I'm really glad to be here in person this year because too many times I have given talks online for you guys. So, thanks again for inviting me. Today, I'm trying to give you an insight on our research about gene therapy and functional biomarkers for drug discovery. But I'll start from the end. Presenting our Italian team. This is a picture that we have taken during the last Walk of Strength on June 15, organized thanks to the newly established Italian association called Creatine, and we did it in Arezzo. Unfortunately, I don't have a picture here, but of course I would like to thank also my, well, not mine, the BCH team. I'm very proud to be part of it during these days. So thank you to Michele and Adeline for the great work we are doing on FNIRS recordings. But I will tell you about it a little bit more later. So our work basically starts always with the mouse model of creatine transporter deficiency that we have generated and fully characterized 10 years ago by now in collaboration with the group of Cornelius Gross at the MBL. This mouse model basically carries the deletion of some exons in the SLC6A8 gene, but the important thing is that basically there is a loss of function of the creatine transporter, and these mice recapitulate very closely the clinical picture of CTD patients, starting with lower levels of creatine in the brain, but also in peripheral tissues, and but also in terms of cognitive function, autistic-like features, the presence of seizures. Indeed, this is a neat map showing that in different behavioral tests that we can conduct in mice, you can see that the knockout animals has an impaired performance, here shown in red. The only better performance is present in the self-grooming and the rotor task, which are basically two different behavioral tasks that we are using for testing the presence of serotype movements. So basically the take-home message is that the mouse model closely mimics the clinical picture of patients, both in terms of cognitive function, autistic-like features, and seizures. So we are now using this mouse model for many different research lines. The first one I want to show you is one that we started in collaboration with the group of Alessandro Gozzi at the Italian Institute of Technology in Genoa. We did with them a characterization of this mouse model with the resting state fMRI that basically allowed us to see what happens between different brain regions in the brain of this mouse model. And what we have found is that there is a strong hypoconnectivity in the brain of these mice. This means that among different brain regions, there is a lower level of connections. Basically, here you can see that there are these blue lines between different brain regions, and the blue code means that the two different regions are communicating less. So basically, these behavioral and imaging readouts serve as a good readouts for testing potential therapeutics. And one that we are working with with much effort is gene therapy. And I'm going to show you the work that did Elsa Ghirardini, which was a former postdoc in my lab. So basically, we cloned the human sequence of SLC6A8 gene under the control of a JET promoter, which is a strong ubiquitous promoter promoting the expression, high levels of expression of a transgene in the mammalian brain. And we also put an HA tag at the C-terminal, so at one side of the gene, SLC6A8 gene, because there is no available antibody for detecting creatine transporter, unfortunately, in the tissue. So we need a tag to see where the creatine transporter is expressed. And so we packed this plasmid into a viral vector, actually an AAV9, about which Jerry was talking before. And we started injecting this viral vector in the ventricles of wild-type and knockout mice very early. The idea is to try to maximize the beneficial effect of our treatment, starting as soon as possible, basically. What ELSA did at first was to collect the brain tissues of these animals after 20 days or 40 days after the treatment to see whether we can detect the real expression of the transgene, so whether this strategy can actually reinstate the expression of the transporter in the brain and also the levels of creatine. What she found, using, of course, the HA tag and using the Western blot, is that actually in the injected brain, there is the expression of the functional gene that we put in the viral vector, while the same band was not present in untreated animals. And this was followed by a significant increase in creatine levels in the knockout mice, both at P20, so 20 days after the treatment, and the same increase is pretty stable and present also at P40, so 40 days after the treatment. And with another technique called immunohistochemistry that basically allows us to map the expression of the transgene in different brain regions, because with the Western blot we basically homogenate the brain and see what happens in the wall tissue, we have seen that there is a good expression, a good transgene biodistribution across different brain regions with a good diffusion across the anterior-posterior axis of the brain. So basically these results prompted us to start a full preclinical trial using this technique, and again we did that in collaboration with Alessandro Gozzi, and we basically injected the animals again with this viral vector, and we performed a longitudinal analysis of the phenotype of the mice at different time points to see the efficacy of our treatment. And we did that using, well, the measure of body weight, because as Jerry mentioned before, this is a very good surrogate of health of animals, but also using the resting state fMRI and different behavioral testing. So many different behavioral readouts. At the end, we measured the creatine levels to see whether the increase that we have seen after 3 weeks or 6 weeks was long-lasting. What we have found first is that our treatment slightly improved the body weight of the animals. You can see that in our mouse model, the body weight is definitely lower with respect to control animals, but there is a slight increase in the animals treated with the viral vector. The slight increase is probably due to the fact that we are not systemically injecting the animals, so we are not affecting the muscle, not affecting the peripheral tissues, but only the brain. So these somehow affect the growth of the animals. But in any case, this is a readout of a better health of these animals. Then I'm going to show you the results of the resting state fMRI, where basically, if you remember before, I showed you that we had hypoconnectivity in the brain of mutant mice. With the treatment with the viral vector, the blue turned out to be red. And this means that comparing animals treated with the viral vector to animals treated with the control viral vector carrying GFP, which is just a fluorescent reported, but not SLC6A8, there is instead an increase in the connectivity among different brain areas. And this means that basically our treatment brings back the communication among different brain areas to the level of the wild-type animals. And this happened, unfortunately, only after 40 days after, only 40 days after the treatment, but this amelioration, this improvement doesn't last along the life of the animals, because at P140, so when the animals are 6 months old, we didn't see any recovery. And And at the behavioral level, we found that our treatment leads to an improvement of autistic-like behavior. This is a self-grooming test in which we basically score the time the animals spend grooming themselves, which is, again, a readout of stereotype movements. As you can see here, the time of grooming is significantly increased in mutant animals, but the treatment with the viral vector strongly reduce this time, so suggesting that there is an amelioration of autistic-like phenotype in these mice. And in contrast, we didn't see any difference, any improvement of the cognitive performance after the treatment of our mouse model. So there is a strong impairment in the cognitive function. Here I'm showing you the results of just one test, basically testing the working memory of these animals, but we have done many different tests and the results are the same. There is no recovery of cognitive function, of the impaired cognitive function of mutant mice with our construct. So as I told you before, at the end of the experimental schedule, we took again the brain samples, and again we found that there was a significant increase in the creatine brain levels, not reaching, of course, the physiological levels that we measured for wild-type animals, but there is a threefold increase with respect to the mutant animals not treated. So this is really significant, but again, this is a measure of the whole brain, and we are not able to map how much creatine we restore in different brain cells. One interesting thing is that, however, that there is a direct correlation between the levels of creatine and the performance in the self-grooming, meaning that the more creatine we can restore in the mouse brain, and the less autistic-like stereotype movements we detected in the animals. That's suggesting that, well, of course, maybe is a message very obvious, but the more creatine we can get in the brain, and the best performance we can expect from these mice. So basically, to conclude this part, these results basically provide a proof of concept demonstration of the feasibility and the potential application of gene therapy for CTD. But of course, we cannot be satisfied with these results because we found just partial beneficial effect and also some safety issues. Maybe we can discuss later about it. I'm not going to show you the results. But the important thing is that we need to optimize, re-engineer our viral vector to obtain maximize the benefits of the treatment and mitigate the potential toxicity. And so this is why I have the luck to work with this wonderful group, and they are now testing a novel construct. We reasoned that probably the lack of complete efficacy of our treatment was due to the impossibility to reproduce the physiological level of wild-type animals. The physiological levels of creatine of wild-type animals in our mutant mice. But we cannot exclude that this is also a matter of cell-specific expression, because from previous results that we published, this is an analysis of the RNA expression of SLC6A8 in different cell population. We know that there is a strong heterogeneity in the expression of SLC6A8 because the brain is made of many different cells, not only neurons, but also astrocytes, microglia, and oligodendrocytes. And it is known also from the work from Stephen in the last years that the expression of the transporter is not the same for each different cell population. We have, for example, some cells that express more and some others that express less. So maybe our work needs to aim to replicate the physiological expression of the transporter and not just increase the levels of expression everywhere. Okay, so this is why we are working on two different novel genetic constructs in collaboration with Stuart Cobb at the University of Edinburgh. I'm not disclosing, the identity of the constructs to protect the patentability of the construct and the future clinical trial. But what I can show you is that actually with one of them in a cellular line model in vitro, we found that with this novel construct, we got lower level of expression of the creatine transporter, again assessed using the HA tag with respect to the previous construct containing the jet promoter. So we hope that this is the right way to produce a more effective gene therapy for CTD. But of course, and we are now getting the new virus to start again a full clinical trial with many, many readouts. You have seen that we have behavioral measures. We can do EEG measures. We can do imaging in collaboration with Alessandro at the IIT. But we always need more objective readouts for the drug discovery to set the validity of a treatment, right? So the second part of my talk will be focused on the importance of assessing the potential benefits of therapies using another biomarker of brain function. And since creatine transporter deficiency and creatine deficiencies in general strongly affect the brain metabolism, we decided to focus on a new technique for the field called the imaging of intrinsic optical signals, because this technique is totally non-invasive and allows to perform a direct measure of the metabolic activity in the brain. So basically, the activity of neurons require more oxygen, more nutrients, basically more metabolism in the brain tissue. And this technique allows us to measure this increase in metabolism during neural activity. And we are focusing on the imaging of visually evoked responses because there is increasing evidence in the literature, partly provided by the work of Michela Fagiolini at BCH, showing that the visual system can be a very good proxy for assessing brain function in neurodevelopmental disorders. So we use the imaging of intrinsic optical signals that Raffaele Mazziotti, which was a former postdoc in the lab, introduced some years ago. And Giulia, which is a postdoc now in the lab, did the work in the CTD mouse model, basically measuring visually evoked signals in wild type and knockout animals at different time points corresponding to different stages of disease progression. And as you can see here, there is a significant difference which is stably present across time between knockout mice and mutant and wild type animals. And even if you can see that the dots, the blue dots representing the mutant mice are lower with respect to the gray white dots representing the wild types, this means that the amplitude of the signals is higher in mutant animals, and this is why imaging of intrinsic signals basically measures the reflectance, the absorbance of the tissue of the red light. So the higher is the amplitude and the lower is the signal that we actually get. And so in addition to this very stable increase in the amplitude of the signal, we also found that in heterozygous female, the amplitude of the signal is basically between the amplitude that we have found in knockout animals and what we have observed in wild types. So suggesting that there is a direct relationship between the levels of SLC6A8 expression and the amplitude of this signal. And another thing that I want to show you is that using PCA analysis with a quite fancy way to see whether a measure can actually segregate mutant animals from wild types, you can see that with respect to EEG, for example, the imaging of intrinsic signal is very effective because you can see that the blue dots are all on the right side while the white triangles are all on the left side. And this means that this measure is very effective in segregating the two different phenotypes, the two different genotypes, and so it's very effective in discriminating between, let's say, knockout mice and wild types. And Giulia also found that there is correlation between the amplitude of the signal and the behavioral performance of the animals. And this is true both for cognitive performance and for autistic-like features. So basically what she found is that the higher is the amplitude of the signal and the poorer is the performance of the animals in the cognitive tests and in the rotarod. So suggesting that this signal can be also predictive of the severity of symptoms. And this is a second very important feature of a biomarker. And the third important feature is that the biomarker should be modulated by a potential therapy. So we tested the biomarker using the cyclocreatine treatment, which is, well, probably you are familiar with that, which is a drug mimicking creatine functions. And what we have found is that animals treated with creatine basically have an amplitude of the signal which is lower with respect with untreated animals. So basically recovering the levels of wild-type animals. And this means that probably the biomarker is modulated by a potential therapeutic. And the same is true with gene therapy, but we are just at the starting point of this experiment. But preliminary results suggest that also with gene therapy we could achieve a decrease in the amplitude of this signal. So for the very last part of this talk, I would like to show you what we are doing in the patients, because of course all of our work in the animal models aims to produce something useful for the patients. And the imaging of intrinsic signal has a very good translational value, because there is this technique called functional near-infrared spectroscopy. I guess that many of you are familiar with that right now, which is a totally non-invasive technique and cup-based, allowing to do in humans the very same measurements that we have done in mice. And well, we set up this system at the Stella Maris Institute with the help of Elena Scaffè, which is a brilliant child neurologist working with me in Italy. And when we started to do that, she immediately realized that the common type of visual stimulation used for assessing visual evoked responses made of a checkerboard and the gray baseline can be very, very boring. So we decided to produce an innovative visual stimulus, merging a cartoon to the classic visual stimulus. And basically this is how our stimulus looks like. There is a cartoon, while with the audio on, you can hear the audio now, but believe me, it's on. And sometimes randomly appears this checkerboard, which represents the actual stimulus. Now the audio is on.
All right, where's the break? You feel a break?
Basically, sometimes this is definitely more entertaining than a gray screen, right? So this will allow us, the children, to see the movie more properly. So we validated, I'm going fast here, we validated this technique in neurotypical subjects, both in adults and children. And what we have found is that basically the cartoon movie, the cartoon stimulus is effective in eliciting a standard visual responses as the classic checkerboard did. And we also found that the response, the amplitude of the response is basically independent from the cartoon chosen and the contrast at which we are showing the cartoon. And this means that basically we can, every single subject can watch the movie or the cartoon he prefers, and the response will be okay. And again, we validated the same in neurotypical children, but let me show you what we are doing in the CTD population right now. Well, the sample size that we have in Italy, because the patients actually diagnosed in Italy are well, the number of patients is very small, so the sample size is not very high. But this is a preliminary study which is very promising, because we found that there is feasibility of this technique both in controls but also in CTD patients. Because as you can see here, the red line is the actual signal and the gray line is the signal to the baseline. So you can see that there is something that we can measure and compare to the amplitude of the controls. This is what we have done in children. This is the same in adults. And this is the comparison, the direct comparison between controls and the CTD patients. And what you can see is that despite the very low number, there is the same tendency towards an increase of the amplitude of the signal, as we have seen in mice. But of course, in Italy, we have so many patients, so our work cannot be enough, and this is why we needed to go multi-site. And well, we started a collaboration first with the group of Aurore Curie in Lyon, getting an European project, and we are looking forward to have their results. And we started a collaboration with the group of Michela Fagiolini here in the US at BCH. And thanks to the great work of ACD and well, the teamwork that we did, we started to do collecting results, well, recordings and data, even here during this symposium. So I would like to thank all, well, of course, Heidi Sanghita and all the ACD Foundation for making this real, but also all the families that already participated or are going to participate to the studies during these days. I would like also to tell you that there are some spots open for Monday afternoon, so if any other want to join, well, we will be very happy. And yeah, basically this is it. Thanks for, well, this is again a list of people working with me and the funding agency, but thank you for listening and your attention.
Wow, that was a great talk, Laura. lots of different areas of excellence. So, any questions for Laura? Yeah.
Thank you. I'm excited for the FNIR study. I have one question on that VIOS testing. Looked like you had different time points. Did you use the same mouse for different time points?
Yes.
Did they do worse over time?
No, actually the study was longitudinal, so we used the same animals for doing 3 different measures, but the difference in the amplitude is the same at the different time points. So we didn't see any worsening in the signal.
My question is more to all 3 presenters. So because Dr. Wilder presented that he had to give tacrolimus and steroid before giving the gene therapy, my question to you and Jerry is, do you have to give any immunosuppression before giving your gene therapy?
Well, for now, with this specific therapy, with the therapy, with the construct I showed you, we also try to add the immunosuppressants in parallel, but we didn't see any difference. For the new virus, we have to see. Probably yes.
This question comes up a lot. I think it depends on what you're trying to do. What's the purpose? For liver-based gene therapy, you will need to, in present day, 2024, will need to give a steroid minimum because the patients typically will develop elevated liver enzymes 8, 12, 16 weeks later, if not before, to blunt the response. If you're trying to develop tolerance, immune tolerance to the transgene, the field is evolving. Some people will say you can do steroids only and not have an effect. Others will say you might want to add steroids, Rapamune, and possibly rituximab for a period of time. But this area is quite in flux. I only know that Barry Byrne at University of Florida is adding rituximab. No one else is doing that in the United States for any trial at present, but I know that people are thinking about it. So I think in the minimum, again, thinking about what you're trying to do, steroids would be a minimum. The other agents, depending on circumstance and the way the field goes.
Yeah, in any case, what we are aiming to do right now is to find the right construct for CTD, and then we'll eventually add some adjuvants to the therapy. But if we don't reproduce the physiological levels of expression of creatine transporter, I think that we have to wait for adding anything else.
Yeah, because even with SMA therapy now, there are people that are rethinking how to give it because, it is not a benign therapy. So many patients have had severe reactions to that one. So that's what I'm saying. I'm saying this simply because for the family to understand that moving from mice to human is not quite a breeze.
No, for sure. We are at the very first step of the preclinical studies. And then the work is very long.
And Jerry, do you know, is the rituximab given before viral therapy or is it after viral therapy? Are they trying to suppress the pre-existing AAV serological response, or are they trying to allow for later dosing?
At present, the thought is to give it before, and it's primarily to prevent an anti-transgene immune response, not an anti-AAV immune response. You'll still have the anti-AAV immune response, unfortunately.
And the humoral response is thought to be affecting the target cell type. I guess, is the cargo extracellular in that case? Do you know?
Well, for example, hemophilia A,
it would be.
Yeah, I think people are generally just giving steroids. So it again comes back, because I've talked a lot about with people about this, it comes back to what you're trying to achieve from the beginning. In this circumstance, for these patients, I completely agree with Laura. We have to just have constructs that work that treat the disorder. In the future, if you're going to give it ICV or intrathecal, you may just need steroids. But if you're trying to, you have a protein that's doesn't express at all, and you're worried about an immune response against a transgene, these might need to be individualized. This is, I've been on a number of DSMBs, data safety monitoring boards, and people are doing it different depending on the circumstances. And I think it would depend on what the mutation is. Certain patients might need different things, but in a trial, you have to take, unfortunately, what's the lowest hanging fruit and make it easiest, because you want to get the drug approved if it's efficacious. So I understand the question, but I think right now,
it's too early.
We need to have the optimal construct. I think one thing I would take away from this is that all of them so far increase creatine, which is great.
Yeah.
And then it's just a matter of functional readouts and what is the best dose and targeting strategy. Yeah.
My question is on, listening to the number of gene therapy talks that we have. There's GAMT and there's SLC6A8. My question is more pertinent for SLC6A8. Is there a common standard that we should, or what would be sort of an aspirational endpoints that we should look at for gene therapy, right? Do we say some level of creatine? If we say behaviors, we've seen a variety of behaviors. There's hyperactivity, there's cognition, there's stereotypic behaviors. Is there something that we can define as a minimum threshold of behavioral correction that we'd like to see with the gene therapy that will give us a little bit more confidence about translation and success? And this is open to all experts in the room.
Yeah, I can start. In my opinion, I'm afraid that it's very difficult to say because you can measure creatine levels, but you cannot have the cellular resolution for measuring creatine levels. So you don't know if you are overexpressing creatine, the creatine transporter, and overloading creatine in some cells, and maybe expressing not enough levels in other cells. So ideally, in my opinion, the ideal readout is a combination of different measures, starting from creatine, but going also to biomarker of brain function and behavioral stuff.
So I was actually getting ready to ask that same question just as to the group in general, is like, what do we think the target creatine levels are? Because I've probably talked to half a dozen to a dozen venture capital companies who are looking to fund this space, and that's the first question they ask me every single time, and I cannot give them a straight answer. I know that at 50% with the heterozygous females, we don't get as strong of a phenotype that we do with the knockout males, obviously. So that's always sort of where I stick with, but I think that it's an important question, and I think that 50% is reasonable, but again, it depends on development. You have to also, when do we hit that 50%? If we hit it at P40, it might be too late. If we hit it at P20, we're probably okay, but in a mouse. So yeah, I mean, I think that's an important question for sure, and I think it's something as a community we do need to think about.
You only have one patient with a milder phenotype. Well, milder phenotype, he has the full-blown phenotype, but then started doing better and responding to therapy, and his activity is about 30% of normal, creatine transporter. So with 30% is not enough to prevent a phenotype. So you need to give more creatine and more transporter activity than 30% to the cell that need it. Now, when you do gene therapy, you give that to cells that need it and cells that, do they need it? I don't know. So, and that it is the problem. So if we can give the therapy to the cell that need it, again, it needs to be 30% or more. In a homogenate, I have no idea.
Yeah.
Yeah.
The problem is biodistribution.
I think that's an excellent point, Nicola, is that 30% is being driven by the endogenous regulatory elements in the cell type that it needs to be in. And when we're giving exogenous virus, it may not be delivered that way. So, yeah, and to that sort of further point, Laura, in the discriminative index that you tested for working memory, I think, in the wild type animals, was there
there is a detrimental effect,
yeah.
Yes. And what we are testing wild type animals because we want to see what happens in patients with residual activity of the transporter. And so they are our test bed for these kind of patients and also for safety issues. With this strategy, we have found detrimental effects in wild-type animals, and this is why I was mentioning safety issues that we want to resolve before doing anything else.
Yeah, I believe that, yeah. No, in excellent design, yeah. Oh.
Just as a response to Dr. Longo's comment there, I think it's really unlikely that we're going to get in any transduced cell with any of the constructs we're using less than over 100% increase in activity. Even if we're using promoters, we're all using promoters based off viral sequences, and even the weakest of these are still pretty strong promoters relative to endogenous promoters. So I think any transduced cell is going to have a pretty high activity. Unless, of course, Laura makes some new ones. But at the moment, all the available promoters are still going to have well above 100% activity in any transduced cell.
Yeah, great points. Okay. Oh, one more.
This is just something that I keep hearing throughout this conference, and I just want to say something about it so that the parents with children don't go home a little depressed. There's a lot of comments about results where the mice are too old and it's too long. It's been too long. It's and there's been other conversations in the hallway about, hey, like, don't we kind of know with GAMT that whenever you get creatine, it helps. And I just want to second that. So if anyone is feeling discouraged with their research or discouraged about their child because they're getting older, the GAMT families I know with patients in their 20s and 30s come back only a couple of months later and say they're doing so much better, and they keep doing better. Yeah, we never get to do this again. That's why we're going for newborn screening. We never get to turn back time, but we can have a better future. We want this cure. We want these treatments, even if, mice. And I think that Jerry's example of his mouse doesn't model GAMT perfectly. Well, maybe CTD mice don't model CTD perfectly, and we're going to see something even better in patients than we're seeing in mice. I just want to share that thought.
I'll actually second that a little bit because the lifespan of mouse is 18 months to 2 years. If it takes us 60 days to put creatine levels back to normal in a mouse, that's going to be a significant portion of the animal's life. But when we see GAMT and we supplement with GAMT, those creatine levels raise at about the same level, about the same rate that we see in the mice. When we give the mice creatine, like the AGAT mice creatine, it is that same level of ramp up. So yeah, you can't compare with what we find in the mouse and the challenges that we have in the mouse because of their shorter lifespan as a direct analogy to humans all the time, right? So that's one of the things too, as you think about this, by the time that those creatine levels hit the full level of a mouse, that mouse has gone from an infant to an adult, as opposed to not even hitting your half birthday, right? So think about that. You just always kind of keep that in context, is that our lifespan is 40 times that of a mouse's, and that makes a huge difference.
That's
sorry,
a great point.
I have an additional comment. Of course, I totally agree with your idea, actually, because we started with the very early gene therapy at P1 just to see if we can maximize the effects. But one of our aims is also to see what happens if we treat the animals later when the symptoms are already manifest. This is something that we can do as soon as we have a construct that works very well early. It would be a waste of work if we are doing the same with this, which is not so satisfactory, right? So, but it's our plan to do also treatment later.
Just one other comment, and there's probably a couple people in the room who could comment about this also. For the families, in arginase deficiency, what I spoke about as an example, I would have been the first one to say the changes that are there are going to be permanent and you can't change them. However, there was a drug, an enzyme, an arginase enzyme that was given to those patients in a clinical trial. And individual patients, their parents, will tell you how much their child improved neurologically. They will tell you. Some of them will tell you this. And then unfortunately, the company the trial didn't meet endpoints. That's part of the reason why the core outcome set is so important. The things that Laura's trying to develop, things that we can measure in trials, whether it's AGAT or it's GAMT or it's CTD, are so important. And the drug didn't get reviewed by the FDA. In the end, most recently, a European company bought the rights to the drug, and the drug got approved in Europe, and it's for those patients. But some of those parents who had older children, teenagers even in their 20s, were improving. Of course, that reversed when they came off the enzyme, and it's hoped that it'll come back. So I'm not trying to give you false hope. I'm trying to tell you I think the brain is more plastic, more changeable than maybe we realize in juveniles and in adulthood. That I would have been the first one to not say that, and now I think there's some evidence, at least anecdotally, to suggest otherwise, at least in one metabolic disorder.
Yeah, we have similar results with cyclocreatine, because starting dosing cyclocreatine during postnatal development, even if the drug didn't get to the clinical trial for other reasons, improve somehow the cognitive function and the autistic-like behavior of the animals. So I think that even late treatment could be somehow working.
I want to second Jerry's comment about the parents noting during the clinical trial that things were improving. I helped lead a clinical trial in a different disease. We made a very big battery to try to measure as many endpoints as possible. But it was a new drug on a new application that had not been tested before. So we were making our best hypotheses. And unfortunately, we had the same experience. The parents were telling us things that were happening in their child that we did not have a measure for, and so it did not hit the endpoints, and it failed. And it has really impressed upon me the need for pilot clinical trials before you make, because once you make the big clinical trial, it's too late if you don't know what's going to happen. So for anybody creating a therapy that is going to be in humans, please do a pilot study. I know it takes longer and there's a little bit of delay, but it might be the difference between what you can end up finding and what you can't. And it's weird, like you think you're gonna know what a medicine does, but in the medicine that we were testing, what we found was that the kids were parents were saying, my child sits with me at dinner now. Used to just go up in the room. This is a severely intellectually disabled kid, not participating in the family because they just can't follow the conversation. They're just not part of it. And now he's less irritable. But in a different way, not captured by the ABC. But he just sits with us, and he's part of the group now. And that wasn't captured in any of our measures. And one of the, in this particular disease, the concept that people thought of was, for this population, it was like they were more serene than they had been before. And that could have been a primary outcome measure if we knew that that might be an outcome. But we didn't, because we didn't have a pilot test, and it's not something that anybody had thought was important to the disease. But it became important once the medicine was being tested. So please consider doing a pilot test. And I've told Ultragenyx and anybody else, like, if you're going to test something, invite a psychologist to go sit. I will go sit at the hospital and watch people take, I will observe, I will interview, just in a very open-ended way to see what is happening, even if it's healthy adults or healthy, adults, either healthy adults or healthy or older patient populations. But it's really, I think it's so sad to hear it's not, and it has happened with several things where there probably was an impact, but we picked the wrong measure because we didn't pilot test it first.
I know we need to go on the break, but I'm going to give a plug for my talk where I'm going to talk about exactly this. But at least for this, we all know that we're going to have open label studies that will be pilot for this. But it's also a plug for the natural history, because you can't just start with these measures in these treatment trials. So more plug for registry and natural history data that will allow us to work on these measures.
Okay, so we'll cut it off. A very stimulating talk, Laura. So, fantastic work.